So I'm very pleased to be here today with you and to share a bit of insight on the project, the Icarus project. Okay, that should work now. Which is about improving crops in arid regions and future climate. We focus on pearl millet and sorghum, which are two plants that are endemic of the Sahel, which are dry regions. So that's one of the few projects that focus on an agriculture problem and a food security issue. And here what we try to do is to find solutions to address the issue of water stress, which is already, of course, an issue now, but which is going to be like even more acute in future climate. So I guess, many of you are not from my own discipline, so my objective and my task today is to make you understand, at least in its broad terms, the idea of the project, and that's going to be like the first part of my talk. And in the second part, the largest part of the talk, I'll give you again, a lot of rationale and progress in the different work package. And the project is in itself, very pluridisciplinary. The first work package is on molecular physiology, the second on ecophysiology and agronomy, the third one on genetics, and the fourth one on crop simulation modeling. So we talk about plants and plant-water relation, and why is it important to talk about temperature in this? So as you may know, plant photosynthesize, so at the level of those leaves that I've represented here, I guess you follow my arrow, no? So,
yes, I can see it.
Okay, so basically there's an exchange of carbon dioxide at the leaf level with water vapor, and that's a necessary trade-off. So in other words, there's no photosynthesis without losing water. And so the case that I've represented in the top of my slide is a case where the atmosphere is fairly wet. And so the gradient that drives the water out of the leaves are not very, very high. And the climate change, that's what's going to happen. The air is going to be like hotter, it's going to be drier, and the gradients for driving water vapor out of the leaves are going to be a lot more important. And that's going to create what we call an atmospheric drought. So to represent those gradients, we talk about evaporative demand, we talk about vapor pressure deficit, VPD. And so I'll be talking about those two terms a lot in the presentation today. And that's basically the center of the project. We want to solve that complicated issue for plants, is how to keep on photosynthesizing without losing too much water in those conditions of water stress, atmospheric for water stress that crops will be facing in the future. So a few years ago, we were, of course, looking at this in the scope of, a project on water stress. And here on that graph, what we see in the y-axis is basically water losses, transpiration, as a function of the evaporative demand, the VPD. Between two lines of pearl millet, one of them, we increase the vapor pressure deficit, transpiration increase linearly. So the atmospheric evaporative demand increases, the plant responds. In the case of this other genotype here, or cultivar if you prefer, where we've got a lower loss already, but past a certain level of vapor pressure deficit, we've got even a further restriction in those water losses. And so we've got two different cases in two different cultivars. For us, it's important because the difference between those two curves represent water savings. So we've got basically a line of pearl millet here that uses a lot of water, and we've got a line here which happen to be drought tolerant. That's why we use them to compare, saves water. So that's basically the basics for the project, and it saves water under high evaporative demand. So we expanded that research on more genotype and more genotype of more species. Here, that's the case of pearl millet, but we did that in many other species. And we came up with different kind of response. So that linear response that you have, we can have also a linear response, but with slopes that are less important. And so, well, that represents also water saving. Or we can have all kind of situation where the slope can be the same, but with a breakpoint in between. All of these situations represent different degree of water savings. And so we start the project with the knowledge of genetic variation for that transpiration response to an increase in evaporative demand. So why is it so important? The benefit number one is that it has a relation with what we call transpiration efficiency. Transpiration efficiency is basically a measurement of how much biomass is produced by a crop, by a plant, per unit of water consumed. So what we did was to take, here that's data that are generated in sorghum. We took the case of two sets of genotype, 10 lines with a low response to evaporative demand, and 10 lines that were insensitive to the increase in evaporative demand. And we measured the water use efficiency. We measured water use efficiency, transpiration efficiency. I'll be alternating between those two terms. We'll consider they are equivalent. What we found is that those that are insensitive to vapor pressure deficit, or those that have the lowest transpiration efficiency. And in contrary, those that were sensitive to the increase in evaporative demand, or those that have the highest water use efficiency. This we measured with a system that I've developed in India. These are called lysimeters, and so you may not very well see on the screen, but they are long tubes where we grow plants all the way through maturity. And every week we come with that sort of a scaffold, and we weigh them, we lift them, and we weigh them, and we measure how much water is used throughout the cropping cycle. So we've got a very, very precise measurement of the water budget. The second, why TE is important is because it relates to yield. And here I've given a few examples of relationship between grain yield here in the y-axis and transpiration efficiency in the x-axis. In the case of maize, in the case of peanuts, in the case of sorghum, I've got an example like this. The second benefit is that since plants use less water under high evaporative demand, they keep that water available for later during the cropping cycle. And that benefits grain feeding. And that's very, very important. Here, if you see the graph, it's cumulated water use as a function of time in two sets of cultivars that are either tolerant or sensitive to water stress. What happens is the tolerant one tend to use a bit less water at a vegetative stage. They use a bit more at the reproductive stage. Overall, both groups use the same amount of water. They simply use it in a different manner throughout the cropping cycle. And if you play a little bit with the data to do regression analysis, if we plot the water use here and plot it against grain yield, we've got a negative relation. If we plot the water use around that stage, plot it against grain yield, we've got a positive relationship. So in other words, the plants that save water at an early stage have more water left for the rest of the cycle, increase their yield because they better feel their grain. So it's a big second benefit. So what we want to do from that, well, we want to understand the mechanism behind that transpiration restriction. We want to understand how it interacts with the environment. We want to find the genetic determinants, and we want to predict where that trait could have the biggest benefit. And that's basically the structure of the project. So in the work package one, we want to understand how it works. So I like to represent a plant as if it was a set of tubes and resistors. Okay. So here, take it that it is a plant. It is a plant, and there's two places of resistance at the root level here, and I've represented a root cylinder here, and at the leaf level, and the leaves here, and the place for resistance is basically the parenchyma and the stomata. Questions that we raise in that project: why do we have that cutoff here? Or why do we have lower slope? Is it related to differences in the plant hydraulics? So when we talk about plant hydraulics, we have to understand that it can be in many different places of the plant that we have that restriction. It could be, first of all, at the level of the soil itself and the soil plant interaction at the level of the rhizosphere. It could be in the root radial anatomy. It could be at the level of plant membranes. We'll talk about aquaporin later, which are water transporters. It could be about the plant axis at the xylem level. It could be in the leaves. It could be in the leaf anatomy. where aquaporine also play a role. So there's many different places in the plant that we need to explore that could be potential candidate to explain those potential transpiration restriction and those potential hydraulic restriction. In the scope of the project, we focus on the root because we've got preliminary evidence that the restriction could take place there. And here is a bit of data that we have already collected, where we compare, those lines that contrast for the transpiration response to VPD, which we have here, contrasting for the root architecture, for the xylem anatomy, contrasting for the root hydraulic conductance. And we've got many more things ongoing. There's an interesting insight also, as I was saying, the soil may also play an important role in that situation. What we did was to measure transpiration efficiency, which is like the integrator of the effect of the transpiration response to VPD. And we found that we had a soil effect on transpiration efficiency. Here we compared in 2 different seasons, a season with a high VPD, a season with a low VPD. We compare transpiration efficiency in 4 soils: an alphy soil, a sandy loam, sand soil, and a vertisoil. So 3 different soils. And in that high VPD season, we see that the sand soil, has a lower TE than in the vertisoil, for example. But in the low VPD season, we don't have any significant difference. What is interesting is what we tested here were 3 species. And here is exactly the same graph where I make a distinction between the species that we tested. So here, it's again transpiration efficiency in those same 4 soils. But as you can see, we've got differences in maize. So maize doesn't have the same transpiration efficiency in those 4 different soils. So that's a black bar. But the white bar of pearl millet, we've got exactly the same transpiration efficiency in the different soils. So what we understand is that those transpiration efficiency differences are caused by the transpiration restriction to VPDs. This, we've got evidence for it. But we realize that we've got a soil by species interaction in that response. And so what it tells us is whenever we do experiment or wherever we do experiment, considering the soil effect is going to be extremely important. For example, when I come with my sets of genotypes that I've tested in India over the years, tested in a very clayey soil like that black soil, and now I'm testing them in Senegal where the soil are extremely sandy, I've got to take into account that if I do take pearl millet, there's probably going to be no problem. But if I take another species, I may have to face interaction issues that definitely I need to take into account. The second part of that first work package is a role for aquaporine in explaining the differences in the root hydraulics. So what are aquaporines? They are water channel transporter, the water transporter. They are, protein with a 4D structure embedded in the plasma membrane, and they facilitate the transport of water. And they do have a very, very important role in water transport pretty much everywhere in the plant, and in particular in those in the root cylinder. Here is an example of where they can play a role. They can play a role in the water transport in the leaves, in the water transport in the roots, in the root cylinder. And here is an example of what happens when we apply an aquaporine inhibitor on the root hydraulic conductance, which we call it PR, with no inhibitor. When we apply the inhibitor, when we restore and we remove the inhibitor and restore the normal conditions. So a strong effect of an inhibitor on aquaporine on root hydraulic conductance. So that's going to be like what the kind of experiment we are doing to test whether we've got different degrees of inhibition of the root hydraulic conductance. The other thing that we've looked at is whether we've got differences in gene expression of aquaporine genes. And here we started doing that with a few lines that were either insensitive to the response in VPD. So this is basically the typical response of transpiration to VPD that we've seen since the beginning of the talk, and a line that was sensitive to the vapor pressure deficit. And here are 2 aquaporine genes that are differentially expressed, and they are more expressed in the line that is insensitive. So in other words, the line that is insensitive tend to have a higher gene expression of aquaporine. And so the hypothesis behind is that assuming that, well, those genes expressed turn into proteins and embedded in membranes, and so we've got more water transport taking place. What we will need to do is to confirm that with more contrasting lines for that transpiration response to VPD. In the work package 2, we want to understand what happened in the long term, because when we do an experiment where we measure transpiration response to VPD, what we do is we put the plant in a growth chamber, we raise the VPD level and we change it every hour, and we come and weigh the pots and we measure transpiration. It takes a day and it's a very transient response. But the thing is, the plant that would have grown in different conditions may have an effect on eventually that response. And we know that we have already this kind of effect. And so what we have to understand is that here we need to better grasp balance between two surfaces of exchange. The root area that are in charge of absorbing water, and the leaf, which is in charge of photosynthesizing, but that's where the water is lost. And so we have to understand better about that balance between two exchange surface takes place. And so here is a situation when the plant is grown under low VPD. Okay, we have a certain balance. If we grow the plant under high VPD, what happens? Do we have the roots remaining intact and the leaves becoming more resistive, less resistive, bigger, larger, smaller? We don't know. Is it the contrary? So, and this, we have very, very limited data on this, and that's something that we need to explore. So what we have been doing, for instance, is compare how the leaf area developed here in the y-axis as a function of the VPD in the growth condition. So we've got here low VPD condition, high VPD condition, and we did that with sorghum, we did that with pearl millet. In sorghum, no change. We apply a high VPD, the leaf area develops equally. In the case of pearl millet, it decreases. So we've got also a species sort of contrast to take into account here. But we know that we've got a VPD effect, at least in pearl millet. What we did also was to measure the root area to leaf area ratio, again in plants that were grown either under high VPD in the red and a low VPD in the blue. So here, that's the root area to leaf area ratio in different genotypes, that's 20. As you can see, by and large, the root area to leaf area ratio increases. I say by and large because in some lines, the increase is very large. In some lines, there's no change. So by and large, plants that are grown under high vapor pressure deficit tend to produce more roots relatively to leaves. So they get prepared to basically channel water to support transpiration, but it doesn't happen the same way in all the genotypes. So that's the genotypic difference that we want to understand, and we want to understand how much it is related to those differences in the transpiration response to VPD. The second part of that second work package is related to another aspect of the project because the project is about water saving, and one of the things that also is a potential water saver is if we intensify cropping, we tend to have the soil less exposed to radiation, and we tend to, we have a chance here to save water. So what we wanted to do in the context of two crops, pearl millet and sorghum here in the Sahel that tend to be like grown more and more at higher density, we wanted to look at the link that we could have with first, in relation to water savings, but could there be a link between the density response and the response of transpiration to VPD in the context of an increase in density? So the thing that we did, that's something that I did a few years ago. We tested the plant response in commercial cultivars of pearl millet. So we measured the yield under high density, and we measured the yield under low density. And here is basically the 1-1 line. So we've got a response here in some lines, but not in all of them. So we know that we've got genetic variation taking place in the response to density. Why is that? We don't know. The reason why I've started that work is if you look at maize breeding over about a century, the planting density of maize about a century ago was 3 plants per square meter, and today it's about 9 plants per square meter. But crops like sorghum and pearl millet have not basically followed the same trend. And so the question here is, why is that, first of all? And is there a way that we can exploit those genetic differences? We have lines that respond a lot to density and some other that don't. Could we exploit those genetic differences to do faster what maize has done over more or less a century of breeding without any deliberate effort to breed for an increase in density? Of course, we increase densities that raise a lot of questions, again, related to that schematic plant. Here is situation under low density. When we put the plant thicker one to the other, again, what happens in terms of root and leaf area ratio? Do we have like the roots becoming smaller? Do they remain the same? And the same for the canopy. What happens to the canopy? So what kind of effect do we have that do have consequences with regard to VPD? So what's up with the VPD in the canopy? How is the water needs? I mean, of course, we work for Sahelian region, water is limited. If we put more plants, we're going to be using more water. Can we do that? What about transpiration efficiency? We may decrease the VPD in the canopy, so we may have an effect on the transpiration efficiency. And then what happens with the transpiration response to VPD? And what happens to those root to leaf area ratio? So regarding VPD, we measured that in a number of situations, in a number of crops, in different situations, in the glass house, in those lysimeters of India, in lysimeters here in Senegal, in a field trial here in Bombay. And basically, the top graph is VPD under under low density. So we've got a higher VPD in the low density stands. That's the case of the red line here, the gray line here, the gray line here. So it happens all the time at different stages of the crop. So we've got a benefit in terms of VPD because the evaporative demand is going to be lower in a high density stand. So that's very, very important. In terms of water budget and biomass response to density, that's basically the water use in the y-axis here as a function of a high density treatment and a low density treatment. Those experiments were done again in lysimeters. And the setup that we used was in the high density treatment, replication was represented by 4 tubes, each of them grown with 1 plant per tube. And in the low density stand, we had 4 tubes also, but only 1 and 2 was planted. So the water use under high density increased by 41%. But the biomass here, which is the y-axis here, the biomass increased 63%. So in other words, biomass increased relatively more under high density than water use. So it means that we've got a benefit in transpiration efficiency. And the same results were obtained in pearl millet. So here are the data in water use efficiency in Y as a function of, again, the low density treatment. Sorry, the order of the treatment are reversed in those graphs. So the high density treatment, higher water use efficiency in the high density, same thing for pearl millet. The benefit about 20%, but we've got to keep in mind that there's genetic variation for that. So in certain line, the benefit is up to 50%, and some line don't have a response. So here we've got also an increase in water use efficiency under high density, which is relatively more in certain genotypes. So there's a scope for breeding for that response. What is very important is, and very exciting, is I told you before that we had a positive response to density, a positive response that was not the same in all genotypes. But what is very exciting is that this increase in density, in response to density, is driven by water use efficiency differences. And what I've plotted in that graph is a ratio of the water use efficiency under high density. And so the more the water use efficiency increases under high density, the more the biomass under high density increases. So this is basically what drives the positive response to density. What we did also was to measure the transpiration response to a proxy of the evaporative demand. We use a reference evapotranspiration. I don't enter in the detail to explain how we do. And we did find in those genotypes that were contrasting for the density response, we found differences in the slope, so some being restricted and some not being restricted. And what is also very interesting is that there's a link between that slope of response to the evaporative demand and transpiration efficiency. So in other words, you've got cultivar here that do have a very limited response to the evaporative demand, and there are those that have the highest water use efficiency, and highest water use efficiency, highest response to density. So in other words, we established a link between the response to density and the transpiration response to VPD in those stands being exposed to different densities. So that's a very, very exciting link, which we still need to understand. Here are the hypotheses to explain that response. So I've represented here the two canopies that either transpire a lot or not much. So this one has more biomass, and it has that high slope of response to the evaporative demand. And here, that's basically those that have the lowest slope. What happened there? Do we have differences in the VPD in the canopy of those two stands? The second thing that we are hypothesizing now is that we've got differences in the depth of light penetration. So we could have that in that canopy here, because of architectural difference in the canopy, we have a better penetration of the light to deeper level in the canopy where the VPD is lower, which is more beneficial for basically converting biomass converting water into biomass. Whereas here, the canopy would be such that the light would not penetrate deep in the canopy, and it would only benefit the leaves that are at the top of the canopy, those that are exposed more to the VPD of the atmosphere, to higher VPD conditions. And that's part of the reason why these would have a lower water use efficiency. And so I'm just now recruiting a PhD student who is going to be basically dealing with comparing, measuring the 3D architecture traits and comparing genotype for those 3D architecture traits. The third work package, there's a student who is about to start on the genetics of climate change traits. So there's a range of traits that we would like to target. I mean, those that are related to that VPD response that we focused on today, and I'll continue delving on this. And of course, I mean, whenever we've got good candidate architecture trait, I mean, the genetics of it would focus on that. So first of all, it's important to frame the questions and the conditions. That's what we have been measuring so far, a transpiration response to VPD in a situation of a growth chamber where everything is controlled, the VPD, the light, everything, but we really can't scale up to the level where you can do genetics. And so what I've developed when I was still in India was a system where we could do that outdoors. So that's what you've got in the picture here at the bottom. It's a set of what we call load cells or balances. And with this, instead of measuring and controlling the VPD each time, we can't, of course, we're outside. We measure those profiles, their transpiration profile in the course of the day, taking the conditions of the site there in India. It's a natural laboratory with naturally high VPD conditions. And so with this, we can generate a lot of profiles, but of course, we need to sort of understand which feature we should extract from those profiles to do genetics on. And so what we have had to do is to develop a pipeline to analyze those data. And so very quickly, I'll go through. That's basically the load cell response profile. So we see the alternance of several days. Here, that's an irrigation. and then the weight sort of drops again as the plant transpire. So we convert that into a raw profile of evapotranspiration in a smooth profile. And the smooth profile, and that's from that stage onward that we can start ranking genotypes. And so the data sets that we have used to develop that pipeline, we had different groups of genotype with well-known behavior. And we wanted to test whether the pipeline was sensitive enough to basically distinguish those different groups, and we did. So I won't expand much on that pipeline, but basically we have developed a pipeline to treat those load cell data. I mean, there's 1,500 load cells available, so that it's a massive amount of data. And we have also basically extracted features to characterize the transpiration response to VPD in the scope of those daily profiles. So to do that phenotyping effort, we'll have access to the platform that I developed when I was still at ICRISAT. I've got a close relationship to them. So that's basically a set of scanners to measure the leaf area. We need to measure that. So basically 3D laser scanner that drives above the crop canopy. And then that set of load cells that you've seen in the previous picture. And I've developed a similar system at a smaller scale at IRD in Montpellier now. The work package 4 is about crop simulation modeling, because as I've told you before, we know that in certain environment, plant growth may change, the root to leaf area ratio may change. Well, in other words, I mean, we are going to have interactions with environmental conditions. So that's where the crop models are very useful to tell us, I mean, where a particular plant characteristic could have an effect and where it could not. And so that's basically the whole idea of the project. And that's also where we are going to have a linkage to breeding and a linkage to testing hypotheses on the ground. So in other words, that's where we're going to be doing experiments. That's part of the reason why I'm here in Senegal at the moment, where I'll be working with 2 stations here on the ground, where we will be testing variants for those VPD response traits. Also in collaboration with a place where I used to be working in ICRISAT in India, and of course in a continuous interaction with the French institution in Montpellier. The thing is to do crop modeling, it's important to have good background data. I mean, it's not like you take a model and then you run it. You need a lot of information, a lot of data to run it. And as it happens, a lot of those data are not available in the West Africa region. So I've got, a postdoc at the moment who is basically like cranking up things to be able to eventually do a crop simulation. The kind of thing that we would like to do is what is described in that graph here. That's the data that come from India, where we would be testing the effect of that transpiration response trait on crop performance, on crop yield. So that's in the case of sorghum. So what you have in the x-axis here is crop yield across a set of time location combination. Each of those data points is basically one experiment, one in silico experiment that we did with the model. And there's about 700 of them in that graph across different stress scenarios. So basically here, we run the model in the regions where sorghum is cultivated. We took weather data from about 20 stations, and we took weather data from about 50 to 70 years of information. So what you have here is a yield that is achieved by a standard cultivar. And what you've got in the y-axis is how that yield changes when we introduce artificially that one trait, transpiration response to VPD. So here you see that the yield benefit is positive in most of the cases, not in all of them, but in most of the cases, and especially so when we are on the left side of the x-axis, which is in those site per year combinations that are poor, where the yield is low. And so it gives us an idea of where it could work, those scenarios that are in red and yellow. and less so when we get to scenarios that are a bit better and when we go towards the right side on the x-axis. So that's the case in India. We had weather data, we had soil data, we had the cultivar data. We could do that. I would like to do the same thing in West Africa, and we need to assemble all those pieces of the puzzle until the time when you can really reliably do modeling. I should have probably told you what is a crop model first, and for those that don't know. So a crop model is basically a process-driven algorithm, a set of equations, put on stack, that represent basically plant production sort of a component. And it takes input, it takes climate input, soil input, crop parameters input, and agronomic management input. And it basically output a bunch of things. The crop dynamics, the crop phenology, so when it flowers, when it matures, when it emerges. It outputs production, of course, and it outputs a number of balances in terms of nutrient, in terms of water balance. And it needs also validation data. The model is basically a prediction. What you need first is really to be sure that the model gives you prediction that are relevant. And so for that, what we did is to assemble databases, so climate databases, so that are gridded, so every 0.5 degrees. It assembled soil databases, and I'm not going to enter into the detail. And basically what the postdoc is doing is to assemble them in the framework of the crop simulation model that we will be using, so that whenever we want to run the simulation, the setup is ready. We need to assemble crop parameters. And as it happened in pearl millet, we have crop parameters, but not for lines that are adapted to West Africa. So we need to generate those data. And that's the kind of work that we've started doing here in Senegal with popular varieties of the location. So we need to generate crop characteristics that we need for the crop model. We need to have agronomic data to validate the model output. Here on the left side of the slide, I'm giving you what I mean by validation. So that's basically something that we do when we predict the yield across a bunch of trials. So each of those data points is an individual trials. And here we measured yield in those different trials that were done in different countries. That's the case of peanuts. And we predicted the yield with a model for peanuts. And so when we've got a good regression, that like what you have here, it means that the model predicts well what the yield will be. Well, we don't have that yet for pearl millet in the region. So that's the kind of data that we need to acquire. And well, there's a collaboration that is currently ongoing with several of the projects that are interested to similar kind of things. And last but not least, we need to share data because it's very time consuming to assemble those data. So there's a small collective of people across INRA and CIRAD and IRD, and the folks here in Israel and Senegal who are, well, we all have like common interest in sharing those data for different purposes. And so we are working on this to have, let's say, a shared set of data that can be put to work. So in summary of my talk, I hope I've been able to convey the message that transpiration restriction under high VPD matters for crop adaptation to water stress and particular so under climate change. We need to better understand it. There's an hydraulic basis to it. The architecture trait is something that has added really to that project because they are responsible for the density response, but it has a link to the transpiration response to VPD. I've shown you that we can measure those traits at a fairly large scale, and thanks to those collaboration with my former institution. And with the crop simulation, once we have set up things, put things in place here in West Africa, we'll be able to predict where they can have the largest effect and how to guide their use. And that's where really the link to breeding takes place. So I would like to thank very much everybody. Of course, the donor for the project, the MOBGA call, the region, Occitanie, who also pledged some money in it, and the University of Montpellier. Other contributors, in kind, that's CRISAT and IRD. and many partners, and especially the people in Serras and Isra here in Senegal, who are going to be like even more important partners in the few years to come. Thank you very much.
Okay, thank you very much. That was very interesting. So everyone, if you have questions, please use the Q&A tool at the bottom right of your screen. I guess I will start with the first question, which is very general. What do you think, how much more yield is possible in general in the regions if you improve just like how many plants are put per square meter, which plant is used, etc. Having all the other resources the same.
Well, I mean, you've seen the graph of the density response, right? So it's going to vary according to genotypes. But what I would say is going to be like a series of small added benefits. So we would increase the density, and we see that we increase the yield in certain genotype up to 30–40%. So that's going to be like the first added benefit. But we see that we don't proportionally use 30–40% more water. So somehow we save some water, and we are going to have a benefit from the added water, the saved water. But we are going to be like reducing the VPD in the canopy, and we are going to be increasing the water use efficiency. And so that's another added benefit. And then we are going to cover the ground and get also a bit of a benefit from the radiation that it doesn't reach the ground and doesn't evaporate soil water. So what I think we are going to have is a series of added benefits, for example, and others that I don't even know what's going to happen. But I mean, what's going to happen, for example, in terms of soil infiltration, soil water infiltration. I mean, if you've got more organic matter in the soil, we probably can expect a much better soil infiltration instead of runoff. So I anticipate that we would have, I mean, direct benefit in terms of yield, but a lot of unexpected, also equally positive benefits.
Mm-hmm. Okay, excellent. We have a question from Manvendra Dubey. So you are focusing on drought response, but how would crops respond to more higher moisture? I mean, you write that in some regions you will also have the moisture increasing, which could also have negative effects, even I also think, right?
I mean, definitely. I mean, that's a very good question and very relevant one. I mean, it's true that we are in a situation of a climate extreme, and so I mean, this year in Dakar, it's raining like hell. You know, it's amazing. I've never seen rain and I came to Senegal many times. I've never seen rain before, and this year I've seen rain like never. So and last year was dry, for example. So it varies a lot, and sometimes you can have an excess, and sometimes you can have enormous deficit. So if we talk about flooding, I mean, that can happen, yes. And I would say flooding would be probably a reverse situation to the one we face. Would probably involve also, a lot of things in aquaporins. I think, what we need to do really is to have a broader approach to everything. I mean, I'm currently discussing with other people in other sort of discipline, people that do climate prediction. And one of that scientists was telling me what we need to do in a place like West Africa is we should not say that, well, okay, we've got drought and blah, 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 and so our crop are going to be like suffering. There are certain years like this year, where we've got a lot of water, and we need to be capable of grasping those benefits. And I think that's what we will need to do. That's exactly what the Australian agriculture does, extremely successfully, to grasp the benefit of the good years.
Okay, I have a question from Amadou Mame. Which crops are more tolerant to water stress, and is it possible to attain food security in West Africa? Also, are farmers involved in your study?
Also what?
Are farmers involved in your study?
Well, we always think of crops like pearl millet and sorghum being like extremely adapted to water stress. And it owes essentially to their shorter duration, smaller leaf area, smaller basically interception surface. Maize is supposed to be less tolerant, but actually it's extremely efficient at using water. So I wouldn't say that. I don't like when we compare, when we want to have like the best of everything. I mean, the most water stress tolerant, it's always a matter of context. So certain species, a certain genotype cultivar will be better in certain environment and worse in others. So it's important to know what fits where the best, and that's basically where the model sort of is able to help us a lot and guide us a lot. Regarding the engagement with farmers, I mean, that's what is exciting with the work we do in Senegal. I mean, it's something that I'm doing in close collaboration with a colleague of mine, who is also, I mean, basically doing that relationship to farmers. So we're actually about to start to do tests in farmers' field of the density effect, because actually that's we don't need to do genetics for that. It's a simple agronomic management change, so we can already apply it.
Okay, Subu, you raised your hand, then removed it again.
Hi, yeah, sorry. And so, Vincent, thanks for the talk, and interesting to see. And actually, you answered part of my question with your response before about talking to farmers. It is going to be basically, do you work with? Is your research aligned with local government policies? For example, in places like Senegal and all that, whether the existing local governments have policies about best practices or alternatives that they're trying, and whether it is aligned with that.
I would say the MOBGA project is relatively pluridisciplinary. It links up, for example, to the CG system and breeding. But I know well enough, I mean, working with the EIB, for example, the Excellence in Breeding platform, I know well enough that it's not because you breed for an improved cultivar that it's going to be benefiting farmers. I mean, there's still a long way to go. And so the MOBGA project cannot pretend that it's aligned with the national policies. But I mean, my presence here in Senegal is not only related to the MOBGA project, it's related to basically the long-term relationship between my institution and Senegal. And that's where we have, I mean, already a better alignment with the national policy. But again, I mean, it's something that needs to be aligned, and I can't tell, I mean, on something else that I'm currently coordinating between the CG system and French institution on climate change issue, but we're exactly on this. We're exactly in this in multi-actor decision sort of issues on how we can take the output from research and take decision from that. So there we touch about on what you say. I mean, how can we align research aspects to taking decision that fits with basically national strategies, basically. I mean, dealing with, for example, underground water, how the model output that I would be generating, how can they inform people that need to manage water at a watershed level to tell, well, use that crop instead of this one, irrigate or don't irrigate. You know, that's where the linkage is necessary, and that we seek for a better alignment, but we are far from there, unfortunately.
Great. If I can ask one more.
No, let's go ahead.
Yeah. So, I mean, you mentioned that you're currently in Senegal, right?
Yeah.
And that's great. And so, I mean, the MOBGA project in the last year and a half, just a different question in terms of the COVID situation. Could you talk a little bit about how that has impacted your research and your project?
Well, it has been a bit of a pain, because when I was in Montpellier when the things came up, and so at the beginning it was basically firefighting with finishing experiment before we were locked down, so of students. That was 2020 and 2020. So of course this year we wanted to go to Senegal and start to run a bunch of experiments which we could not. So we had to postpone everything for 2020. We managed to send the students, the postdoc, to Senegal. I couldn't come. And that's the reason why I pushed to come, in fact, to be here. And because at least being inside Senegal, I mean, the COVID was not as acute as it was in Europe. And the folks that were here could function. And so now I'm here, I can function, and in case there's a relapse, at least I'll be able to go to the field.
Great, thank you.
Thank you.
Okay, excellent. I maybe have another question. You look a lot on the yield. I wonder if if the fruit to leaf ratio is also something that you are looking at, because I mean, even in marine science, we sometimes have it that we have carbon overconsumption, which means that the algae are just doing photosynthesis and can't even deal with what they are doing. And I wonder if there's something to optimize. And the other question is maybe also, are in some plants there that also where other parts of the plant are also more useful than plant A than plant B? I mean, that they are used for building houses or
yes,
for as a fuel or something like that.
Yeah, I mean, that's an excellent question and a very important one. I mean, again, I mean, we are here dealing with smallholder farming, and it's true that maybe I've sort of used too much yield, grain yield. That's definitely not the focus. But okay, we need to simplify it, I mean, one way or another. Yes, of course, it's very important to think of crop residues also. I mean, here we've got a mixed crop livestock farming system. So, of course, people need grain for food, but they badly need feed. And it's true that we are in the Sahel in situations where the production of feed for cattle is, well, it's an increasing gap. I mean, which put pressure on natural resources, on natural parcours, rangelands. And so for us, I mean, the focus is really, let's produce as much biomass as we can. It's going to be beneficial. It's going to be beneficial because part of that will be converted into grain, because part of that is going to be consumed by the cattle. And in the end, because part of that will be fed back to the soil. I mean, we are in extremely rapid sort of turnover sort of environment in terms of organic matter. But producing biomass is, I think, very, very important here. Yeah. So we have to see all those dimensions of plants optimizing biomass production.
Mm-hmm. Okay, and what about like socio-economic impacts? If you would change like to one crop throughout large areas, you would probably also change a lot in the social system, etc. So, are you considering those things?
Well, not in the Moboga project, maybe beyond in,
I mean, in your
field. In my field, that's something that we've got, I mean, close in mind. Yes, it's very important. But we've got also to realize that I mean, the ag system here is in transition also. I mean, people want to change. I mean, there's a dramatic change. I mean, occurring at the moment. So, yeah.